Among biology topics, photosynthesis and cellular respiration can be very complex.
You'll be required to learn inputs, outputs, and other important factors of each of the processes.
To help clarify these processes for you, we have created a short and entertaining film about students who, like you, are about to learn all about how cells make and use energy. Hannibal.
Hey, you're late.
That's okay, I have chocolate. There. Yep. There you go.
Thanks.
Yes.
Hey, what's this? What's this?
That's a golden ticket. Whoa.
For the glucose factory?
Yes.
Let's go.
Let's go. Yeah.
Hello students, today we're going to learn about the processes of photosynthesis and cellular respiration. Come into the factory. Right now we are in the cytosol and we are about to enter the chloroplast. Our first stop is the thylakoid, a structure inside of the chloroplast containing light-absorbing pigments called chlorophyll. Do you know what happens here?
Something with light?
The light reactions.
Right, let's go! If you look over there, you can see light system 2.
Don't you mean light system 1?
No, light system 2 comes before light system 1. They were named for the order in which they were discovered.
Oh, that makes sense.
Light energy enters photosystem 2, where it jumps across chlorophyll pigments and is finally accepted by the primary electron donor P680. Breaking apart a water molecule provides P680 with an electron, which is excited by the light energy and sent to the primary electron acceptor. The protein PQ then transports the electron to the cytochrome complex. With the electron, the cytochrome complex will generate ATP. This process uses an ADP and a phosphorus. Then the protein PC will transport the electrons to P700 section of photosystem 1. Meanwhile, more light energy is being absorbed, sent across the chlorophyll to the P700 section, exciting the electron and sending it to the primary acceptor, where the protein FD will take the electron and send it to enzyme NADP+. Enzyme NADP+ will use the electron to reduce NADP+, which is an electron carrier. The electron turns it into NADPH. This will be used later in oxidative phosphorylation. That's all for the light reactions. Next we'll see the Calvin cycle. I would like you to meet a very special enzyme named Rubisco. Do you know what Rubisco does?
It performs carbon fixation.
Correct! Let's see Rubisco work. Rubisco receives a 5-carbon molecule of RUBP and fixes another carbon to it. The now unstable molecule breaks in half. ATP donate phosphorus to each molecule, then electron carriers and NADPH donate electrons, becoming oxidized to NADP+, and creating 2G3P molecules. Once 4 more are made, 1 is donated to glycolysis and the other 5 are turned into RUBP. RUBP is sent back to Rubisco. NADP+, ADP, and sugar are gained from this process. Now the Calvin cycle can begin again and photosynthesis is concluded. Let's go see cellular respiration now. Complex reactions out here in the cytosol have turned the G3P you saw in the Calvin cycle into glucose. And the cytosol is also where you will see the process of glycolysis, where glucose is broken down into 2 pyruvate molecules. If you look over there, you can see glycolysis taking place. This process does not just split glucose in half. Many intricate reactions take place to control the energy released, as glucose is split into 2 pyruvates.
Look at all those complex reactions. Do we have to know all of that?
You don't need to know the specific reactions, just know the net gains. 2 NADH and 2 ATP are gained. Do you know what comes next?
The Krebs cycle.
Right, now we will see the citric acid cycle, also known as the Krebs cycle. The Krebs cycle takes place in the cell's mitochondria. This level of the mitochondria is called the mitochondrial matrix. Active transport takes a pyruvate to a multi-enzyme complex which begins to reorganize the molecule. In the process, an electron is released which is used to reduce NAD+ electron carrier into NADH. Once the molecule is converted into acetyl-CoA, it enters the cycle. The CoA is broken off, followed by 2CO2. Then, 3 NAD+ electron carriers each take an electron from the molecule and are reduced into NADH. ADP and phosphorus are then combined through interaction with the molecule to make ATP. Then another electron carrier, FAD+, is reduced by one of the molecule's electrons. Leftover components are returned to the beginning of the cycle. NADH, FADH2, ATP, and CO2 are gained. The cycle will repeat when new supplies arrive from glycolysis. Electronegative electron-transporting proteins span the mitochondrial membrane. Note the high hydrogen ion concentration on this side. The electron carrier NADH drops off its electrons at the beginning of the chain. The electron then travels down to more electronegative components. The electron carrier FADH drops off electrons at a lower point on the chain, meaning less energy will be gained from it. Do you know what receives the electrons at the end of the chain?
Is it oxygen?
Yes, it is. Since oxygen is extremely electronegative, it can take electrons from the end of the chain. The oxygen then combines with hydrogen, making water.
Shouldn't this process make ATP?
That happens in the next part of oxidative phosphorylation, called chemiosmosis. You see, when the electrons travel down the electron transport chain, they bind to oxygen and then to hydrogen at the bottom, creating water. That leaves less hydrogen ions down there and more hydrogen ions up here, which creates a proton gradient. Let's go see the ATP synthase elevator. The protein ATP synthase is powered by hydrogen ions moving down the proton gradient. This movement allows ATP synthase to combine ADP and phosphorus to create ATP. Hydrogen ions enter ATP synthase. When they do so, they cause a part of the protein to spin. The motion allows the protein to create ATP by combining ADP and phosphorus. This step gives 90% of the ATP generated by cellular respiration. (30 to 34 ATP are gained. And that's all for cellular respiration. I hope you learned a lot here at the glucose factory.
Hey, Naval, we're sure gonna pass that test later. Test?
In conclusion, photosynthesis consists of light reactions in the Calvin cycle.
Cell respiration starts with glycolysis and then moves on to the Krebs cycle and then finally ends with oxidative phosphorylation where most of the energy is made.
To sum it up, we have this table of inputs and outputs for the different stages.
Good luck learning biology!